let's get started with section 4 of chapter 9, the inheritance and genetics chapter. So here in section 4, what we're going to be looking at is basically how nature breaks all of Mendel's rules, the stuff that we just finished talking about in the last two videos. So the thing is that a lot of genes do work in the way we just talked about, where you've got dominant and recessive genes, and you get the homozygous dominant, homozygous recessive, heterozygous, and so you've got basically just the dominant version of the phenotype and the recessive version of the phenotype. But it's not always so straightforward. There are exceptions to Mendel's rules. So that's what we're going to be looking at here. So the first set that we're going to look at is what we call incomplete dominance and codominance. So this is where we get effects of both alleles showing up in the phenotype. So there's no true dominant and no true recessive gene in these two exceptions. So let's take a look at each of these. We're going to look at incomplete dominance first. Now, incomplete dominance is where when you get the heterozygote, so the one that has both copies, is actually a blending of the two parents. So we can see that the most common one that we see this in is in flowers. So you can see we start with a red snapdragon, a red flower, and a white flower, a white snapdragon. And when these two are crossed together, what we get, instead of getting red or white, what we end up with are pink snapdragons. So all of the offspring are pink. They each have a white and a red allele, and the two mix together to make pink. If you take the offspring, the two pink flowers, and breed them together, then you'll get white flowers, pink flowers, and red flowers. So all of the genes are still there. It's just with incomplete dominance, neither gene, neither the white nor the red, is completely dominant over the other. And so when you get them together, they mix together in order to make that new color, the third option, basically. So the opposite of this is codominance. And this is where you get the heterozygote, the heterozygous version, displaying the characteristics of both homozygous genes. So I like this picture. It's not completely true, but it's a kind of a nice way to think about it. So if you think of mom and dad, dad having the horizontal stripes, mom having the vertical stripes, codominance would be plaid, because you get horizontal and vertical stripes. Both of them show up in the offspring. So let's take a look at how this works with actual genes. So in codominance, this is where the heterozygous alleles are both expressed, no blending. So in our cow over here, for example, our cow is the fur color. So in our cow, the fur is white and brown. So those are the two genes. So they're not blended together. If this was incomplete dominance, then the cow would be kind of a light brown color. Instead, what we get is spots. And so this is an example of codominance. An example of codominance, kind of like the plaid in humans, is our blood types, specifically the AB part of our blood types. And so if you look here, we've got people who have type A. A is just referring to these little flags. These are protein flags that are identifying that blood, those blood cells, as being your blood cells. B-type blood, you get B flags. Okay, so if you have homozygous A, or if you are heterozygous, meaning that you have A and then O, that's what the little I is, then you're going to get A flags. Same for B, same sort of type. We've got homozygous B, we've got heterozygous, you've got B and an O. The codominance comes in here when you get, let me move me out of the way again, when you get A and B, when you get the A allele and the B allele. When you inherit those, you get codominance. You get both A flags and B flags on those red blood cells. So that's where the codominance part comes in for the AB blood type. So let's stick with blood types because blood types actually give us a couple more basically breaking the rules for Mendel. So for Mendel is just looking at one allele, two alleles. So A or B, short or tall, purple or white. Mendel got really lucky with P's because there are only 2 alleles for each of the things he looked at. But some genes have more than 2 alleles. There's more than 2 choices, and blood is one of them. So multiple allelism is where a single gene has more than 2 alleles, but each individual still only carries 2 alleles. So you still only get 2 copies. So this is where you get that variation. So the best example of this is the ABO blood group in humans. So the alleles are A, B, and O. So for blood, there are 3 alleles. And for A and B, they are, and you may have caught this when I said it earlier, A and B are dominant to O. And so it's sometimes written as little I, like you saw in the last slide. And so whenever you get, if you inherit an A allele with an O allele, then the A is going to be dominant. Same with the B. And then we just saw A and B are codominant to each other. So if they're inherited together, you are going to be AB, because both of them are going to get expressed. So this gets us 4 blood types, A, B, AB, and O. So those are the 4 major blood types. Now you might be thinking, but I've also heard of like AB positive. I'll get to that here in a little bit because that's actually a different set of genes. So let's take a look at those gene combinations for the blood types. So this is written out a little differently than what we saw there on the other page. So this is just with the letters. So you can see here, if you have type A blood, you can have homozygous dominant, so 2 copies of the A, or you can be heterozygous, where A is dominant to O. So you can have AO. So type A people can have both types. Now I'm type B, so I could either be BB, so homozygous, or I could be BO, I could be heterozygous. And I actually don't know which one I am. Likely I am homozygous because both of my parents have B blood types and so does my brother. So if you do Punnett squares for blood types, the likelihood that all 4 of us have B blood types shows that likely this is where my blood type is at. For AB, only one option because A and B are dominant or are codominant to each other. And then O, the only way you get O blood type is because you've got homozygous recessive, because you have to have the 2 recessive genes to have the O blood type. So why is it then if you get the wrong blood type, it can be so dangerous? Well, this is why. When you inherit your alleles for your antigens, these protein flags that are on your blood cells, you don't just inherit the gene that makes these. You also inherit the gene that makes antibodies. Okay, so you get these antigens, these flags on your blood cells, and you get these B antibodies if you have type A. Now, if you look, these guys are going to they won't fit. They're the wrong color to attach to A antigens. So they won't attack your own red blood cells, but if you get type B cells into your system, they will attack. Okay, and that actually can set off a huge immune response, and it can, if it's severe enough, actually cause death. You've got the reverse for type B. So people who have type B blood make A antibodies, so they would attack type A. People who have AB blood, no antibodies, because if they did, they would attack their own blood. So they don't have any antibodies. And then people who have type O, they have no antigens. So they make both types of antibodies because they have no flags, so they want to keep an eye out for anybody who has a flag. So if they get any of these guys in, they're going to go and attack them. So is this information important to know? Yes, because there are types of blood that people can and can't get. So for example, like I said, I'm type B. So if I need blood, there are only two types of blood that I can get. I can only get type B, like my own, because it would have those B antigens that my body won't attack. Or I can get type O, because there's nothing for my body to attack on type O. There's no flags, no A antigens, no A flags to go after, so type O is safe for someone like myself with type B blood. Type A blood, same sort of thing. They can get type A, they can get type O. But if you look down here, got a little different story for the other two blood types. Type AB, these guys can get blood from anybody because they have no antibodies. They have both types of flags, so they can get all four blood types because their body won't attack anything. Very different for type O. Type O can only get type O because remember, they have A and B antibodies, and so they can only get blood cells that don't have those flags on them. And so this is one of the big reasons why when you go into the hospital, if you need blood, they're going to type your blood to make sure they get the best match. Now, as far as donating, you can see a little different. You can donate, if you're type A, to anybody who has an A allele. If you're type B, you can donate to anybody who has a B allele. If you're type AB, you can only donate to AB blood types because they have those flags. Anybody else, they would attack those blood cells. Type O, because they have no flags, can again donate to anybody like we saw over here because they have no markers. Now, I told you I was going to talk about that other part, the positive negative part. So positive negative is another gene. It's the RH marker. It's a single gene that just has two alleles. So it's either there or not. So this is not an example of multiple allelism. Just the ABO part of your blood is. So this is just an either or. If you have one or two copies of the RH marker, so basically other flags, you're Rh positive. If you're an individual with recessive alleles for this Rh marker, you're going to be Rh negative. So that's what that positive negative on your blood type is it's these extra markers that show up on your blood. All right, so the next lecture is going to pick up with multi-gene traits that are actually going to continue on. We talked about multiple alleles. Now we're going to talk about multi-gene, adding genes together in our next lecture.